Hello everyone, this lecture video is all about pentose phosphate pathway or PPP. We can define this as a metabolic pathway by which the glucose 6-phosphate is used to produce the reduced form of nicotinamide adenine dinucleotide phosphate and ribose 5-phosphate, and as alternative pathway for the production of intermediates in glycolysis and amino acid synthesis. So take note that the main goal of this, one of the main goal is to produce the NADPH and not the NADH. Okay, that is different. So later you will know what's the importance of this one. So this is significant in cells, that is cells that produces lipids such as in fatty tissue, liver, mammary glands, and the adrenal cortex. So this adrenal cortex is essential in the production of steroids, which occurs in amino acids and for hormones as well. And then the major functions for PPP is the synthesis of the reduced form of nicotinamide adenine dinucleotide phosphate, then also the production of ribose 5-phosphate, which is being used in DNA, RNA, then also the production of pentose derivative needed for the synthesis of nucleic acids, among others. So, just like what I have defined earlier, this pentose phosphate utilizes the glucose 6-phosphate as an alternative pathway for the production of other important enzymes in our body or other important molecules in our body. This is where it will take place here in glucose 6-phosphate. So as I have mentioned, instead of NAD and NADH, so one of the goals of this pathway is to utilize the oxidized form of nicotinamide adenine dinucleotide phosphate into its reduced form, which is the nicotinamide adenine dinucleotide phosphate hydrogen. Okay. And then this pentose phosphate pathway has two stages. First is the oxidative stage, in which there is a loss of electron. So this is the first stage for oxidative, this one. So this is the second step, and this is the third step. So first, we have here the glucose 6-phosphate, which will be converted into 6-phosphogluconolactone with the help of the enzyme glucose 6-phosphate dehydrogenase. And it will utilize the oxidized form, which will be converted later into reduced form of NADPH. So the hydrogen here will be removed since this is a dehydrogenase, and you will have a double bond here, and you will now have the 6-phosphogluconolactone. Then the second stage, we will have here the enzyme 6-phosphoglucolactonase, which will stop the binding in the site, and now you will have here the 6-phosphoglucanate, this one. And when that happens, a water molecule will be released. And then the third step is that the 6-phosphoglucanate here, with the help of the enzyme 6-phosphogluconate dehydrogenase, and with the help of this high energy compound, it will be converted into the reduced form, and this one will be removed, the carboxylate ion here, and it will produce a carbon dioxide gas. Then you will now have the ribulose 5-phosphate. This ribulose 5-phosphate here will now proceed on the non-oxidative stage here. This ribulose 5-phosphate will be isomerized with the help of the enzyme ribulose 5-phosphate isomerase. So this one will be isomerized in this way. And now you have the ribose 5-phosphate. This ribose 5-phosphate is essential in the as a backbone, one of the backbones of DNA and RNA, and in the production of ATP, is the adenosine triphosphate, guanine triphosphate, uridine triphosphate, coenzyme A, oxidized form of nicotinamide adenine dinucleotide, and the reduced form of nicotinamide adenine dinucleotide. It is also essential in the production of oxidized form of flavine adenine dinucleotide and the reduced form of flavine adenine dinucleotide. Then, after that, so if there is abundant production of ribose 5-phosphate, some of the will react to cellulose 5-phosphate. So when it will react to the cellulose 5-phosphate, it will produce the glyceraldehyde 3-phosphate, which is this one is essential in Krebs cycle in glycolysis. And it will also produce the pseudo-heftulose 7-phosphate. Then these two here, this glyceraldehyde and the pseudo-heftulose 7-phosphate will be interconverted with each other, and they will produce the fructose 6-phosphate and erythrose 4-phosphate. So this fructose 6-phosphate is essential in the glycolysis, and the erythrose 4-phosphate serves as precursor in amino acids production. Then, one of the functions of the reduced form is in glutathione. So, glutathione is the main antioxidant used by the body to keep the hemoglobin in its reduced form. So, to keep this one going, the body needs the reduced form for the regeneration of the depleted glutathione, which is this one is being continuously being used in the hemoglobin. So, ensuing lack of regeneration of glutathione leads to the destruction of hemoglobin-containing red blood cells. And when this happens, a severe anemia will occur, which will make you feel weak and tired as always, and you will have a difficulty in pregnancy, and you will continuously feel pain. Then severe anemia can lead to the irregular heartbeat, which is the arrhythmia. So that is why the production of the reduced form of nicotinamide adenine dinucleotide phosphate is essential in our body, along with the production of ribose 5-phosphate and erythrose 4-phosphate as in amino acids, and the fructose 6-phosphate, which this one is an alternative pathway for the production or regeneration of this one. And then that is all for the pentose phosphate pathway.